So today we will be solving the questions from Unit 13, and also we will be learning the strategy of how to solve a particular type of question. So first of all, I will be giving you a few questions to solve. Okay, like first of all, you have to solve the question, and then I'll give you around 2 to 3 minutes to solve the question, and after that I will tell you about the correct answer and the explanation of that particular question. So are you all ready? Can I get some yes in the chat? Okay, I can see many students are, I think they are trying to log in because, so I'll start the class in a minute. Let's join, everybody join. Okay, so let's begin with the first question. So please go through the question and tell me the answer. The question is about molecular markers. Which one of the following statements regarding molecular markers for genotyping is correct? So four options are, genotyping is incorrect, and four options are given to you. Polymorphism in intronic regions of a gene cannot be used for trait mapping. Second option is codominant molecular markers can be used to detect heterozygosity. Option third is sequence tagged microsatellite sites and simple sequence repeat polymorphisms are based on polymorphisms in repetitive DNA sequences. And fourth option is restriction fragment length polymorphism and simple sequence repeats are multi-allelic markers. So read the question carefully, and I'm giving you around 3 minutes to solve this question. Read the question carefully, and this question you can only answer if you are thorough with the basic concepts and the terms. Like you should know what are codominant molecular markers, what is polymorphism. You need to know about these molecular markers which are mentioned in this question: RFLP, SSR, STM. And when you are reading the question, make sure that you read these words correctly. I know many students, they go through the question, but they simply they don't focus on these words like correct or incorrect, true or not true. So even if you know the question and you don't read the question carefully, if you know the answer and you don't read the question carefully, then there is a possibility you can do a major mistake while solving the question. So in any question, if such words are given, correct, incorrect, true, not true, ascending, descending, please read it carefully. So yes, what is the answer over here? So let's first understand the question. So question is about the molecular markers that Which of the following statements regarding molecular markers for genotyping is incorrect? Now, what is genotyping basically? Genotyping, it is the process of determining differences in genetic makeup of an individual by examining individual's DNA sequence and comparing it to another individual sequence or reference sequence. Now, 4 options are given to you. So you have to find out which of the statement is incorrect regarding molecular markers for genotyping. So first statement is that polymorphism in intronic regions of a gene cannot be used for trait mapping. I'm sure you all know about introns, right? So introns are the non-coding regions within a gene and they do not code for any protein, but they can contain important regulatory elements and variations. What kind of variations? Like single nucleotide polymorphisms, deletions, insertions, okay? And even other variations might occur in intronic region. So basically, the question arises why one need to study about the intronic region. So as I told you that many variations can occur in this region. So by comparing the presence or absence of specific intronic polymorphism with expression of particular trait, researchers can conduct association studies. So the statement is that polymorphism in intronic regions of a gene cannot be used for trait mapping. So this statement is incorrect. Polymorphism, as I just like I told you a few minutes back, that polymorphism in intronic regions definitely can be used. By comparing the presence or absence of specific intronic polymorphism with expression of particular trait, researchers, they can conduct association studies. So yes, they use it for trait mapping. So statement 1 is incorrect. Second statement is codominant markers can be used to detect heterozygosity. Now, what are codominant markers? See, there are 2 terms. One is dominant marker and another is codominant markers. Please remember that codominant markers are those markers which can differentiate between the homozygous and heterozygous. Okay, that is, they provide you information whether an individual is homozygous or the genotype is homozygous or heterozygous condition. However, dominant markers, they only tell you about the dominant allele. Okay, like for example, if you're talking about A allele, let's say we're talking about A gene. So A gene can have 2 allele. According to Mendel concept, one is dominant, which we express with uppercase letter, and another, the recessive allele, which we express with lowercase letter. Now, the individual can be either homozygous A, that is having same allele in homozygous condition. So this is homozygous dominant condition, this is homozygous recessive. Or maybe individual can have the 2 different alleles for the same gene. So here, this is heterozygous condition. Now, I told you dominant markers only tell you about dominant allele. So you can see dominant allele is present in homozygous dominant condition also and heterozygous condition also. So using dominant markers, you cannot differentiate between homozygous and heterozygotes. Like here you can see, if we are considering B gene, then we are getting a band in homozygous dominant condition also and heterozygous also. So this is a dominant marker. But if you see codominant marker, it is showing the bands, 2 bands. So this is of progeny, right? P1 is one parent, P2 is another parent, F1 is the progeny, right? Similarly here, P1, P2 parents and F1 is the progeny. So codominant marker, if we are using it, we'll get a band for P1, in P1 also, in P2 also. And because as the progeny inherits the gene from the parents, we should get 2 bands if the marker is codominant. It will tell you about both homozygous and heterozygous condition. But here you can see the band in the recessive, like the band in the, if like P2 is having the homozygous recessive condition, the band is missing over here, right? So dominant markers, they don't differentiate between homozygous and heterozygous. So question is, the statement is codominant molecular markers can be used to detect heterozygosity. So yes, this statement is correct. Because as I have explained you, we can, using codominant markers, differentiate between homozygotes and heterozygotes. Then coming to statement 3. So statement 3 is sequence tagged microsatellite sites and simple sequence repeat polymorphisms are based on polymorphism in repetitive DNA sequence. So those who don't know about STM and SSR, like basically they are microsatellites. Microsatellites are they are the sequences, you can say they are repeating, like sequences which are repeated multiple times, okay, in the genome of an organism. Now the sequence is repeated is only 2 to 3 base pair, but it is repeated multiple times, okay. So basically polymorphic loci present in nuclear DNA and organelle DNA that consist of repeating units of 1 to 10 base pair, mostly 2 to 3 base pair in length. For example, CT. Now this is dinucleotide, right? So basically CT can be repeated multiple times. So maybe in individual 1 it is repeated 5 times, and in individual 2 let's say it is repeated only 3 times. So by using the we use PCR based method here, so we can design the primers and we can amplify that part. So using this we can even determine the polymorphism. So this statement is also correct. We can determine the polymorphism on basis of repetitive DNA sequences, as in STM and SSR. And the advantage of using these markers is because they are evenly distributed throughout the genome. Then RFLP, that stands for restriction fragment length polymorphism. So RFLP basically, again, it helps in detecting the polymorphism on the basis of presence or absence of restriction sites. And SSR, just now I told you. So the statement is that they are multi-allelic markers. So yes, again, this statement is correct. Multi-allelic markers means they are able to detect 3 or more variant alleles for a gene. Multi-allele means when a gene is having 3 or more variant forms. Okay. So yes, we can use, and they are multi-allelic markers. So because in the question, the incorrect statement is asked. So answer is option 1, statement 1. So I hope you all have answered it correctly. Then coming to this question. Now this question is from gene cloning and recombinant DNA technology. So please go through the question. I'm giving you some time to solve and you can tell me your answer and then I'll explain the correct answer to you all. So read the question carefully. Focus on information given in the question. Figure is also given in this question, so see it carefully. Like the options are in form of figure. So first of all, read the question carefully, go through the information given in the question, draw the diagram yourself, and just match it with the options given. The question is, a protein D is encoded by a gene which is 5 kb long and has 3 HIND3 restriction enzyme sites. The first one is 0.5 kb from transcription start site. Second one is 2.5 kb from first site, and third one is 0.5 kb internal to stop codon. So please draw it, okay, because you cannot answer this question without drawing the diagram of the gene, and you have to label the sites, restriction sites for HIND3 restriction enzyme. Then another information given is, so first information is about the restriction sites of HIND3. Second is that second site is polymorphic. The second restriction site for HIND3 is polymorphic. Polymorphic means that variation can occur in this site. So if the site is present in mutated form, it will not be present, right? So in order to find out which fetal cell contain normal or mutated genes, so normal ones will have at the second site, second site will be present, but in mutated gene, it will be lost. So total genomic DNA from fetal cells was isolated and you digest it with HIND3. Now, completely digested means that whatever sites for restriction enzyme HIND3 is present, all will be digested. And then you separate it in agarose gel. Transfer to membrane and then you detect by a probe against the region. Now, this information is again important. Probe you are using against the region between second and third restriction site. So which one of the following band patterns will be obtained if the fetal cell is heterozygous? So draw the diagram and match it with the options. The question appears to be difficult, but it's very easy if you know the strategy of solving the question. I have explained you the question. So what answer you all are getting? Please let me know. Yes, is it option 1, 2, 3 or 4? So let's see, based on information given to us. So the gene is 5 kb long, right? So there is a protein D encoded by gene which is 5 kb long and has 3 HIND3 restriction site. So the first is 0.5 kb. So if I draw it like this, it is 5 kb or 5000 base pair. The first one is 0.5 kb from transcription start site. So if this is the start site, the first site for HIND3 will be over here, right? And this much part is 0.5 kb. Then second one is 2.5 kb from the first site. So this is first site. So from here to here, it will be 2.5 kb. So this is your second site. So this is first, this is second. And third one is 0.5 kb internal to stop codon. So if the stop codon is over here, so internal to it over here, let's say, this is your third site which is present. And from here to here, it will be 0.5. So you have around like 0.5, 0.5, 2.5. So total gene is 5 kb. So how much is this region? It will be 1.5 because this much is 2.5 plus 0.5 plus 0.5 is 3.5. So this much remaining is 1.5 kb. Now it is mentioned that this second site is polymorphic. So in normal gene, it will be present, but in mutated, it will be absent. So you have to draw the diagram for both normal as well as for mutated. Okay. So here you can see this is the normal and the mutated condition, right? So heterozygous condition is like as mentioned in the question that we have to find out, like if you see here, band patterns if the fetal cell is heterozygous. So that means it will have one normal allele and another mutated. So in normal allele, all the three sites for HindIII restriction enzyme is present, but for mutated, second is polymorphic, so it will not be present, right? So that means there will be no digestion occurring at the second site. So second site is polymorphic, the fetal cells were isolated, digested with HindIII, separated in agarose gel and transferred to membrane. And then we are using a probe against the region between second and third restriction site. This information is already given to us in the question. So you can see this is our first site as I've drawn in the previous slide. This is second and this is third. So probe is between second and third. Okay. So probe will bind here and in mutated also probe will bind here. But if you perform the digestion, the HindIII will cut here, it will cut here and it will cut here, right? So that means you will get one fragment of 0.5, another fragment of 2.5, third fragment of 1.5 and fourth fragment of 0.5 kb. But as now you are separating the fragments, transferring it to the membrane and using a probe specifically for second and third restriction site, only the region to which probe will bind will only be seen. Okay. So as in the mutated, second site is missing. So enzyme will cut here and enzyme will cut here. So we will get one fragment of 0.5, another fragment of 4 kb and last of 0.5 kb. But again, when you are using a probe, it will bind only to the region of 4 kb. So always keep in mind that when you are finding the answer, keep in mind that you don't have to tell the fragments generated simply by digestion, simply by complete digestion of the gene with HindIII. You have to keep in mind that we are using a probe and to whatever region probe will bind, only that one will be seen in the end. So for normal, we should get 1.5 kb, right? And for mutated, we should get 4 kb. Why? Because probe is binding here. So only this portion will be seen for mutated. For normal, it will be 1.5 only. So if the fetal cells are heterozygous, we should get one band for 4 kb and another of 1.5 kb, which is given in only option 2, and that will be your answer. So there are small points that you have to keep in mind. Many students, they simply ignore this information that the probe is binding. And they simply, based on complete digestion by HindIII enzyme, they'll solve the question and find the answer. But in that case, your answer will be incorrect. Okay, I hope it is clear to all. Then moving over to the third question, which is again from the gene cloning. And here you can see this question is about vector and the restriction enzyme. So go through the question and tell me the answer over here. Which one of the following combinations of enzymes used for cloning a linear insert fragment into a digested plasmid vector would have the least probability of generating self-ligated vectors in cloning experiment? Following complete digestion of all vector molecules and no further enzymatic treatment of the vector. So here you should know what is the meaning of self-ligated vector, and even the concept related to how we can avoid the formation of self-ligated vector need to be clear here. So 4 options are given to you, and you have to find out the option, the combination of the enzymes used for insert and vector, which will not generate self-ligated vectors, or have the least probability of generating self-ligated vectors, including experiment. So here in the options, if you see carefully, some enzymes, they are generating blunt ends, some are generating sticky ends. So some generate blunt ends, some generate sticky ends. So that also you have to keep in mind while solving the question. So in cloning, what we do? We have a DNA fragment with a gene of our interest. We introduce it into a vector. So you have to perform restriction digestion of your vector. Similarly, if you have a genomic DNA, you will break it into, you will perform the restriction digestion, and you generate several fragments, and whichever fragment with your gene of interest, now you ligate it to your vector, right? And that results into the recombinant DNA molecule. So vector is here is a vehicle which will be carrying your gene of interest, and it has a capability to replicate inside the host cell, right? So let's solve the question. First of all, you need to know about the enzymes which generate blunt ends and sticky ends. Like the concept related to blunt end and sticky end should be clear to you. So when we talk about restriction enzymes, for example, let's say ALU1. So here you can see this ALU1 recognizes 4 base pair sequence, right? And it just cut in middle of the recognition sequence. So for ALU1, the recognition sequence is 4 base pair. And you can see that it cuts just in middle of the recognition sequence. So these type of ends which are generated are called as blunt ends. However, if we take an example of EcoR1, which is another enzyme, restriction enzyme, EcoR1, it cleaves in one of the strand over here. Okay, so it recognizes 6 base pair sequence, GAATTC. Now in one strand, it cuts over here, and on another strand, it cuts 4 to 5 base pair away from it, right? So this kind of ends which are generated with the overhang region, you can see this part is single stranded. We call it as sticky ends or cohesive ends. Why cohesive or sticky ends? Because they stick back by transient base pairing. You can see E can pair with T, right? T can pair with A, and so on. Now in the question, it is mentioned that which of the combination which is shown in the options can prevent the self-ligation, like the least probability of self-ligation. So self-ligation means where the vector will join back to itself without carrying the insert. That is called as self-ligation, where the vector is not carrying your insert, it simply joins back to itself. Now to avoid the self-ligation, 3 things can be done. One is you digest the DNA vector and insert, double digestion of vector and insert. Second is dephosphorylation of 5' ends of vectors. So basically at the 5' end, the phosphate group is present. Okay. So basically what happens in dephosphorylation, what happens is this phosphate group can be removed. So this process is dephosphorylation, because for joining, like phosphodiester bond is formed, right, between the 5' phosphate of one nucleotide to the 3' OH group of another nucleotide. So when you perform dephosphorylation, this phosphate group at the 5' end is removed. Similarly, you can replace 2' deoxyribose with 2' 3' dideoxyribose sugar. So that means OH group is not present at 3' position. So this way what will happen? The self-ligation will not occur, because at 5' end if you remove the phosphate, no phosphodiester bond will be formed, or if you remove the 3' OH group, no phosphodiester bond will be formed. Now, coming to the cloning part. So always remember, blunt end cloning is prone to vector self-ligation. So this is usually the observation of the researchers, that blunt end cloning is prone to self-ligation, because blunt end cloning doesn't have any kind of single and protrusion, like single and overhang regions are not there, and they can simply be ligated to one another. And there are many enzymes which generate blunt ends, like we have SMA1 and we have EcoR5, right? So if both ends need to be ligated into vectors are blunt ended, and we want to prevent the self ligation, vector needs to be dephosphorylated. But if we go through the question, it is clearly mentioned that no any kind of enzymatic treatment is not done, right? So as mentioned in the question that there is no further enzymatic treatment of the vector after digestion, therefore chances of self-ligation will be more in option 1 and 2. We have to find out the least probability. So if you see here option 1, it is, you can see SMA1 generates blunt end. If you see option 2 again, you can see here we have blunt ends, right? The enzyme generate blunt ends. And as we are not doing any kind of enzymatic treatment, so we cannot say that here we have the least probability. Yes, the chances of self-ligation is more over here in option 1 and 2. Now let's have a look at 3 and 4. So that means answer must be between option 3 and 4. Okay, it will not be between 1 and 2. Now if we see option 4, the enzyme is EcoR1, which generates sticky ends, right? You can see GAATTC. And if you see option 3, we have HIND3 XHO1, and vector is also cut with HIND3 and XHO1. So because in option 4 we are using the enzyme which generates sticky ends, so as I told you, the sticky ends, they can stick back by transient base pairing. So it can also lead to self-ligation, right? So for the insertion of a DNA fragment into plasmid vector, please note this is a very important concept, that it is preferable to use 2 different restriction enzymes to digest the DNA, so that different ends are generated, as you can see in option 3. So if we are cutting with HIND3 and XHO1, different ends are generated, and because of that, there will be the least probability of self-ligation. So 2 ends, 2 different ends which are generated, because see here you have the end which is being cut with HIND3, another with XHO1. So obviously there are 2 different ends. You can see the sequences are not same. So because of that, the 2 different ends generated will prevent the religation of the vector without any insert. And therefore, as we have to find out the option which, like the option, the combination, the enzymes which results into least probability of generating self-ligated vector in cloning experiment, answer here is option 3. So you can see in such minute concepts, the like your superficial study for CSIR NET is not work out. Okay, it will not work. You have to thorough with your, you need to thorough with the basic concepts. Your basic concepts need to be very much clear. So answer here is option 3. Then moving over to another question. This question is from Immunotechniques. Following statements are about the features of immunoassays used to assay biomolecules. Radioimmunoassays. So option, like if you go through the statements, statement A is radioimmunoassays are more sensitive than ELISA with chromogenic substrates. Statement B is ELISA with chromogenic substrates are more sensitive than ELISA with chemiluminogenic substrates. C is ELISA spot measures number of cells capable of secreting particular biomolecules using a substrate that gives soluble product with enzyme reaction. And D is that in Western blot analysis, the product of enzyme substrate reaction localizes at the site precisely where antibody enzyme conjugate binds to its specific protein bed. So, which of the following options represents the combination of all correct statements? So to answer this question, you need to be thorough with the technique radioimmunoassay, enzyme-linked immunosorbent assay. You should know what happens in ELISA spot. So principle of these techniques need to be very much clear, even Western blot analysis. So I'm giving you 2 minutes to solve this question. Please go through it and tell me the answer. Read all the statements carefully and you have to find the combination of correct statements, not incorrect. So 1 more minute I'm giving you all. So in CSIR NET, you can get different types of questions. You can get you can get the match the following type question where the techniques can be given to you and maybe their description like principle or application might be given and you have to find the correct combination. Then maybe you can get a statement based question, any figure based question, and even the direct question on a particular technique, principle and application of particular technique you can get in exam. So let's go through statement given one by one. So statement A is that radioimmunoassays are more sensitive than ELISA. So first of all, let me quickly tell you about the principle of RIA and ELISA. RIA is radioimmunoassay. So first of all, RIA and ELISA both are immunological technique. That is where we use antibodies to detect a specific antigen. Now in radioimmunoassay, we code the antibodies for a specific antigen to a well. Okay, so multi-well plate you can take. So let's say you are detecting hepatitis B antigen. Okay, you want to detect hepatitis B antigen in the patient. So what you do, you collect the sample from patient, the blood sample, so you process the blood and you get the serum. And So to detect hepatitis B antigen, you attach the antibody specific for hepatitis B antigen. And now to this plate, you will now be adding some standard hepatitis B antigens which you have in your lab. So it's like a reference antigens, you can say, but they are radio labeled with iodine 125. Now what will happen? These antigens, they will bind to the antibodies, right? And now, so these antigens are radio labeled, okay, with iodine 125. Now, when you add the patient sample and if it is infected, if the patient is infected, like here you can see if it is infected, uninfected, no antigen will be there. But if it is infected, it will have the hepatitis B antigen. And when you add them, when you add the patient sample, these antigens in the patient sample will compete with the radio labeled one, our standard antigens, and will displace them. So when you measure the radioactivity of the complex, obviously here you can see in case when the patient is not infected, the radioactivity will be 100%, right? Because there is no unlabeled antigen, right? Because antigen present in patient sample is unlabeled. We have not labeled it. The reference antigen, we have labeled it. So radioisotopes, they emit radiations, right? The radioactivity which we can measure through some instruments like scintillation counter. So if the patient is infected, the hepatitis B antigens, they compete with the radio labeled one. And you can see that if you measure the radioactivity of this complex now, because unlabeled antigens are here, the radioactivity will be reduced with respect to the one when the patient is uninfected. However, in ELISA, instead of using radioisotopes, we use enzyme. So again here, we use this technique to detect antigens or antibodies or proteins in the sample. So here you can see an example of direct ELISA, where the antibody is specific for the antigen we use. So again, here we use a multi-well plate. We add the patient sample containing antigen. You add the antibody specific for it, and that antibody is conjugated with the enzyme. Now you add a substrate, so enzyme catalyzes the conversion of substrate into colored product. So here, if in the statements mentioned, radio immunoassay is more sensitive or ELISA. So always remember that when we talk about sensitivity, okay, so first of all, let me tell you, as I mentioned a few minutes back, that ELISA, it detects antigen antibody complex, but here we use enzyme. But in RIA, we use radioisotope, right? So when we talk about sensitivity, what is the meaning of sensitivity? Sensitivity technique is sensitive. It means that it can detect very low quantity of your antigen in the sample or whatever you are looking for, your analyte. So here this table, it is, here you can see the sensitivity of all the assays are given. So if you see the radio immunoassay and ELISA, so we can clearly see that ELISA is less sensitive. Okay, so statement A is RIA are more sensitive or enzyme linked immunosorbent assay are more sensitive. So we have to find out correct or incorrect statement. And similarly, you can compare that whether ELISA using chemiluminescence is more sensitive or the simply, so chemiluminescence here it means where the light is being emitted in chemical reaction as a result of chemical reaction. So yes, chemiluminescence, ELISA using chemiluminescence is, you can see, more sensitive in comparison to simple ELISA. Then coming to third statement, so coming back to AB again. So ELISA with chromogenic substrates are more sensitive than ELISA with chemiluminogenic. So yes, chemiluminogenic will be more sensitive. So this statement is incorrect. Okay, A is correct. Then coming to C, ELISA spot. So basically ELISA spot, here you can see an example of ELISA spot where we want to find out whether like the statement mentioned is that it measures the number of cells capable of secreting particular biomolecules using a substrate that gives soluble product with enzyme reaction. So Every word is important. So if we look at the process here, let's say we want to find out cytokines. So we add the anti-cytokine antibody, we add the cells which secrete cytokines. So you have to provide a stimulus, and in presence of stimulus, those cells which secrete cytokine will bind to the antibody. You discard any cell which is not bind, will be washed off, and then you add the another antibody, anti-cytokine antibody, and that is enzyme linked. And then you add the substrate, chromogenic substrate. So enzyme catalyze the conversion of substrate into colored product. So here it is not the soluble product that you get an insoluble product. A blue-black colored precipitate will form at the site of cytokine localization. So this statement C is incorrect. And coming to the last statement, that in Western blot analysis, the product of enzyme substrate reaction localizes at the site precisely where antibody enzyme conjugate binds to its specific protein band. So Western blot, it's a technique which we use to analyze the proteins in the sample to look to find out a particular protein, whether it is there in a sample or not. So here you can see the overview of the technique, that you isolate the proteins from the sample, let's say a cell or a tissue. Now there is not a single protein in the cell, many proteins can be there. So you separate them by performing SDS polyacrylamide gel electrophoresis. The proteins you transfer from gel to the membrane, that is you perform blotting. And finally, you add the antibody conjugated with enzyme specific for your protein of interest or antigen of interest. So antibody will bind to the antigen, right? And you add the substrate. So enzyme catalyze the conversion of substrate into the color. So yes, when you see this sheet, which can be made up of nitrocellulose or PVDF, the antibody enzyme conjugate bind to its specific protein band. So yes, the product of enzyme substrate reaction localize at the site. So this statement is correct. And answer here is therefore option 2. Now moving over to another question. This question is from flow cytometry. During apoptosis, phosphatidylserine, usually present in the inner leaflet of the plasma membrane, flips to outer membrane. Annexin 5 is a protein that binds to phosphatidylserine. Using this as a tool, we identify the apoptotic cells from necrotic and normal cell population by FACS using FITC tagged annexin 5. Propydium iodide is used to stain the nucleus, which generally identifies necrotic and late apoptotic cells. So in which area of the plot you should get early apoptotic cells by FACS analysis. So you can see a diagram is also given to us in the question. So read the question carefully. Although this question is on flow cytometry, but it's a combination question where the concept from the your apoptotic and apoptosis and necrosis, right? This concept is combined with the technique flow cytometry. So based on information given to us in the question, go through it and tell me what is the correct answer here. So you have to find out that which of the quadrants, 1, 2, 3, and 4 out of the quadrants given, which will tell you about the early apoptotic cells. So FACS is fluorescence activated cell sorting. It's an advancement in conventional flow cytometry technique. So usually flow cytometry technique we use to identify a particular cell by using a marker, that is an antigen on its cell surface. We can even determine the size and granularity, like we can even identify the cells based on multiple parameters. We can identify the cell by analyzing its size, granularity. But additional feature, which is the advancement, that even you can sort the cells. Like if you have a mixture of cells, let's say you have mixture of immune cells, B cell, T cell, natural killer cells, so you can separate them in different tubes. So this advancement in conventional flow cytometry is called as FACS, fluorescence activated cell sorting. And from exam point of view also, this FACS and flow cytometry is important, especially the dot plot analysis, the data analysis is very important here. Pay attention to the details. Annexin 5 binds to PS. And it is mentioned that during apoptosis, PS, that is phosphatidylserine, which is usually present in inner leaflet of plasma membrane, flips to outer membrane. So here you can see this is a normal cell. So in normal cell, it is present, phosphatidylserine is present in inner leaflet, right? But when the cell undergo apoptosis, it flips to the outer. And to this annexin 5 can bind. So annexin 5 staining is a very common method for detecting apoptotic cells. They have high affinity for phosphatidylserine. So when the phosphatidylserine, in case of apoptotic cells, when it is exposed on the outer leaflet of the cell, then annexin 5 will bind to it. And it is considered to be very quick and reliable detection method for studying externalization of phosphatidylserine. That is when phosphatidylserine flips from inner leaflet to outer, and it's an indicator of intermediate stage of apoptosis. So in the question, this diagram is given to us, right? So you can see we have FITC on x-axis, PI on y-axis. So you should know these dyes we have used to label what. So we have used the annexin 5, which is tagged with FITC, fluorescein isothiocyanate. And propodium iodide is used to stain the nucleus, which identify necrotic and late apoptotic cell. So I'll tell you a trick over here. Just as we have a number scale, right? Like you have a 0, we consider as a reference point, right? So any values beyond 0 are always positive, and any value beyond, like below 0, will we consider it to be negative, right? So similarly here, I'll give you a trick. What you have to do is, you have 4 quadrants given. So just consider to be a reference point, okay? Now you can see FITC fluorescence, okay? FITC dye fluorescence is increasing left to right. So always remember that this quadrant will indicate you the presence of FITC. So plus here means presence. And this quadrant will indicate absence of FITC. So as I told you, considered to be a 0. So any values beyond 0 is plus, below 0 is minus. So here, this is a reference point which we are taking 0. So beyond this, it will indicate presence of FITC, below absence. Then if I'm considering this y-axis here as a reference point, this one will indicate the absence and this one will indicate presence. So that means this quadrant will indicate absence of PI, right? You can see this is minus, and this one will also indicate absence. Now here if you see quadrant 4, this one will show presence of both FITC and propodium iodide also, both, right? Because you can see here and even here for FITC. And this one, this quadrant 1 will be showing absence of FITC, right? Because for FITC it is absence over here, right? And presence over here. So this one will be presence of propodium iodide but absence of FITC. And we call these quadrants, this one as double negative and this one as quadrant 4 as double positive. So this is how we analyze the data of the flow cytometry. Now, since it is mentioned in the question that annexin 5 is used to identify apoptotic cells. So where we are getting signal for FITC? In quadrant 3 and even in quadrant 4, right? And propodium iodide is used to stain nucleus which identify both necrotic and late apoptotic cell. So that means if we want to identify early apoptotic cell, we should get only signal for FITC and not for propodium iodide. So if you see quadrant 3 is just showing this result. If you see quadrant 4 here, propodium iodide is also there. So that means this one is for early apoptotic cell And this one is for late apoptotic cell, right? Late, and then finally you have the necrosis happening, right? So that means answer here is quadrant 3. So apoptotic cells are always propydium iodide negative and annexin 5 positive. So this is all about the flow cytometry technique. Then we have one more question. So let's go through this question quickly. Absorption spectra of L-tyrosine in acidic, which is shown with continuous line, and basic, which is shown with dotted line, medium was estimated and plotted on a graph as depicted below. So following interpretations were made. Change in the pH from acidic to basic results in shift in lowest energy absorption maxima and decrease in molar absorptivity. Shifting of the absorption band to longer wavelength signifies a shift to lower energy known as redshift. Third statement is shifting of absorption band to shorter wavelength signifies a shift to higher energy known as blue shift. And D's wavelength shift is always accompanied by change in intensity of absorption band. You have to find the correct interpretation. Is it AB, AC, BC, or BD? So I know this question, when it comes, you may find it quite complicated. So here I would like to share a trick. Please remember, this question is from spectroscopy. So always remember a spectrum is given to you. So you don't have to think out of the box. You always have to answer the question based on information given in the question. So we have to go through all the statements one by one. So statement A is that the pH, like the statement A is change in pH from acidic to basic results in shift in lower energy absorption maxima. So you can see the wavelength is given on x-axis and absorbance is given on y-axis, right? And decrease in molar absorptivity. So basically in spectroscopy, we have the concept where like different absorption shifts are observed: hypochromic, bathochromic, hyperchromic, okay? So basically, as you can see here, if there is an increase in wavelength, like maybe due to addition, like if any auxochrome is attached, so that results into change in the intensity of absorption of light. So whether it is resulting into hypsochromic, that is decrease in wavelength, we call it as blue shift, or longer wavelength, we call it as red shift, or is there increase in molar absorptivity coefficient, then we call it as hyperchromic, or it results into decrease, we call it as hypochromic. So in the statement, it is mentioned that because of change in pH from acid to basic, there is a shift in lowest energy absorption maxima and decrease in molar absorptivity. So this interpretation is incorrect, right? Because here the given information does not provide any information on change in lowest energy absorption maxima, right? The graph only provides the information, like if you see here, the dotted and the dashed line is there. So dotted line is basic and continuous line is acidic, right? So if we look here, there is a change definitely in the wavelength, but there is like does not show the absorption spectra in acidic, only shows the absorption spectra in acidic and basic medium, but it doesn't indicate the position of lowest energy absorption maxima. So this statement is incorrect. Coming to B statement, so shifting of absorption band to longer wavelength signifies shift to lower energy. So this statement is correct. Because as you can see here, that when the absorption bands shift to longer wavelength, as in this diagram, we call it as red shift or bathochromic shift, right? So any term can be given to you in the question. So the absorption band, which is shown with the dotted line, you can see it appears at a longer wavelength. Like if I see the peak, it is around 270 here, but it is around beyond 270, right? So it is appearing at a longer wavelength compared to the one. So the absorption band in the basic medium appears at longer wavelength compared to acidic medium, which is shown with the continuous line. So this is correct. Then quickly coming to statement C, shifting of the absorption band to shorter wavelength signifies a shift to higher energy. So basically, we all know that the relation between wavelength and energy is inversely proportional. Higher the energy, shorter the wavelength. So this is a well-known fact. Now, this statement is also correct, although in the given graph, it doesn't show any kind of blue shift. Okay, it talks about red shift, it shows about red shift, but not blue shift. But this statement, because the options given to us is like that, where we have given a two statements, right? Like A and B or B and C, like that. So C is also correct. And D is incorrect, where it states that wavelength shift is always accompanied by change in intensity of absorption band. So here this given graph does not provide any information about change in intensity of absorption band. It is only illustrating shift in absorption band position, right? Which we have already discussed with the change in wavelength, right? So therefore here answer is option 3. So your option 3 is B and C. We have to find the correct interpretation. So your B is correct and even C statement is correct. So this is all for now. I hope you have learned something new today. You have learned the strategy of solving the questions, and all the very best. Keep on solving the questions. We'll see you in another session, and we'll practice some more questions from Unit 13. Thank you, everyone.